Do you want to turn the light down just a little
Sure.
bit? You don't have to turn them both off. That should be good. Thanks. Okay, so last week, Chris spoke to you about the structural pipelines for HCP and what some of the rationale for why they made the decisions that they did with respect to the structural. And so what I'm going to do today is talk about the functional pipelines and both present the methods that they use as well as some of the rationale behind some of the choices that they made. But you can get a lot more detail on those from the Smith, I think it's either 2013 or 2015 paper that's online in the module. Okay. So last time, as I said, Chris talked to you about the structural pipeline and the diffusion pipeline. And so what I'm going to do is focus on the functional pipeline and talk about the pre-processing approaches that they use, but also talk about the steps that come after, which include some of the denoising and just a hint of some of the tools that you could use for analysis. And so this is a little bit of a different take than what you've been hearing throughout the course in terms of using, say, AFNI to do all of your pre-processing. Now, the HCP put together their protocol based on the Siemens scanners, specifically for the Prisma. And we're fortunate that we have been able to replicate their parameters here. And so if you look at the far right panel, you can see that our parameters very largely match up pretty well with what the HCP has proposed. These are a little bit different than what you've heard about for ABCD. Are you familiar with that project at all? Yeah? Okay. Which also uses a similar approach in terms of their data acquisition. Now, for functional imaging, what the HCP is recommending, beyond collecting these T1 weighted and T2 weighted structural images in the diffusion, they also suggest that you use multiband image acquisition on a 32 channel or better head coil. They have suggestions that the resolution of your slice thickness should be less than 2.6 millimeter. Your TR, your repetition time, should be fairly rapid, so a second or less. They also recommend collecting field maps. If you have the ability to do a gradient distortion map, they also recommend having these, as well as a single band reference image. Now, that doesn't mean you can't use their pipeline if you don't follow their recommendations. It is possible to take data that you may have from a prior study and run it through, but you just have to be aware that your results aren't going to be as pristine as what they get. And you may run into some problems along the way in terms of whether some of the decision points that are made within the script work for your data set. So you've heard about this, I think, in one of the first lectures at the beginning of the course, but as I mentioned, the HCP recommends what we call multiband acquisition. Now, many of you who've done imaging up till now are probably pretty familiar with the single band acquisition. That's historically the way that we've gone about our data collection. And the idea is simply that we collect one slice at a time. We typically do an ascending interleaved, which means that we collect, let's see, do I have a mouse? Yes. We collect the odd number slices first, and then we collect the even number slices afterward. And the multiband is a little bit different because instead of collecting these slices individually, we're now collecting a group of slices simultaneously. And so in this example, although we have the same number of slices, as the previous image here, so this has 30, and we're collecting 30 here, we're collecting them essentially 10 at a time. And so it allows us to collect our slices more rapidly than we would with the single band. it also gives us the opportunity to have a faster temporal resolution, as well as increasing our changing the spatial samples that we can have thinner slices. Most of the head coils where you can do multiband have at least 20 channels. Ours here is 32. You'll hear some sites and some of the newer scanners have 64 channel head coil. The downside of multiband acquisition, if you've ever seen these head coils, is that they're a little bit smaller than the single band coil. Yes?
We now have a 32 channel head coil that is as big as 8 channel. So once that's available,
Nice. Yes. So, but at least up till now,
yeah.
I've been corrected. Up till now, it's been difficult. If you have participants with a large head, you aren't necessarily able to use the 32 channel scanner with coil with them. So my sample tends to be adolescents. It's far less of a problem, but in other studies we've run into this with adult samples. So what you can see here is just a comparison between collecting the data on a single band relative to the HCP protocol multiband and the ABCD protocol multiband. And what you'll notice is that there's almost a doubling of the number of slices that we can acquire. So we're improving our spatial resolution because now we're down to about 2 millimeter slice thickness. Our TR is much more rapid. So now we're getting much better temporal resolution as well. The ABCD is a little more coarse in terms of their spatial resolution, a little bit fewer in terms of the number of slices and how many bands they collect. And I think that's really a function of them collecting on multiple sites on multiple scanners. And so this is a way that they have been able to kind of allow them to collect data on Siemens, on GE, and Philips as well. And I'm involved with a study that's using essentially this protocol up at Seattle. And we've been processing with the HCP, and it seems to be working pretty well for us so far. Now, why do you care about your image resolution? One of the things that you've heard about with HCP is the focus on surface-based mapping. And by having these thinner slices, your slices are essentially thinner than your cortical thickness. So now you're kind of getting away from some of that partial volume effect that you otherwise would hit if you're collecting a 3 millimeter slice. This is also helpful in terms of some of the steps we do later on. There's an algorithm called FIX, which helps with denoising your data. It's also helpful with some of the analyses that you might choose to do if you're using these protocols in terms of doing multivariate analysis. So as we know, fMRI is also susceptible to spatial distortion. And this is an example using a left-right phase encoding from the HCP, and you can see that the image is being pulled over in one direction. So you've got the distortion in this phase, as opposed to if you were collecting it right-left, right? You see the pulling in the opposite direction. And so the idea behind the field map is simply to try and correct for some of that distortion. The HCP recommends using a spin echo style simply because it's faster to acquire, which means it's less sensitive to motion. If you've used some of the older field maps and you have a participant move during the field map, now you can't apply it to your functional unless you acquire a new set of field maps. And so I just wanted to show you an example of one subject that was collected here, and what the images look like when we're collecting it on the 8 channel using what would be our standard approach with a 3 millimeter slice thickness, versus if we were to collect it using the HCP protocol and then again with the ABCD, as well as showing you what it looks like when you don't apply this field map correction versus when you do. And what you'll notice is, for one, the multiband, you can see that there's a lot more, because you have improved spatial resolution, we're actually starting to see that this looks more like a brain and a little less like a bright blob, right? And when we apply these field maps, we have less of that pulled banana shape, right? So for the protocol, the project that I've been using, we've been using this functional acquisition specifically. Now, you've also talked about gradient distortion, and the HCP recommends, if you can, to do some sort of gradient distortion correction. And the idea behind the gradient is that because it's not perfectly linear as we would like, you get some distortion. And this is just, I thought this was a cute example because you know what Duplo is. It's like big Lego. And what's really nice is you can see that it's shifted the shape. It's kind of pulled it out a little bit. And the idea behind having some sort of a gradient map that you can then use within the software is it helps with correcting the gradients so that now we're looking a little more like Duplo, and that hasn't been stepped on, I guess. And so the HCP recommends ideally that you do your correction through software. And what's nice is that because they're on the Siemens, they've actually got access to the data file that's needed. It's in the right format for them to actually apply it to the images as they're being processed. For us, it's a little different. That information is considered proprietary by some companies, so we don't have easy access to it, and the files that we are able to get are not in the format that the HCP takes. Now, somebody did write a script that will convert the GE over. However, that script has disappeared. It's no longer available on GitHub, and although the HCP scripts will say, oh yes, we can do distortion correction for GE, it's not really the case because we can't provide it with the right file format for them to actually do what we need to do. But there is a way around it for us. We can actually choose to do the correction at the scanner, which seems to help. This is an example on a structural, but there's this little couple of checkboxes down here, this 3D geometry correction. We found that if we turn that on, it does a reasonably good job, that it minimizes the distortion that we're getting. Okay, so a single band reference image. What they're talking about here is that with the multiband images, because you're requiring them so rapidly, there's less time for T1 relaxation. So sometimes there's not quite as good of a tissue contrast, and having an image that's collected on the single band that allows for this relaxation to occur gives us a sharper contrast that we can then use for the alignment process. So what they basically suggest is that you collect this single band image, same parameters as your functional in terms of the slice thickness. This provides you with sufficient detail for your registration process. And it's used for the purpose of motion correction, so that's the image that we end up aligning all of our other volumes to. It's also used for alignment with our T1 weighted image. If, like me, you don't collect one, you can use the first volume of the time series. And you've probably talked about the pros and cons to that. With, FSL historically chooses the middle volume with the argument that by choosing the middle, there's probably less overall motion to correct for. unless that person makes a big old head bob right in the beginning, then that's going to mess things up. But the first volume, because you haven't reached the full, what's the word I'm looking for? Full magnetization, that tends to have a little bit of a better contrast for doing these alignments too.
So Amanda, when
Yes.
you collect your, on the MR 750 design, we do acquire single band images. You have to do to reconstruct the multiband. Do you just?
So
put it
we
on
haven't,
for you, or?
are those embedded within the multiband?
They can be, or depending on which protocol you're using, they're either done as a separate scan or they're embedded.
I don't think we have a
You don't
separate
have
scan.
access to
We
it?
do not have a separate scan, that I know.
So if you want, would you want to have those images? So you-
Eventually.
Okay,
Yeah, I
so-
mean, we're in the middle of our study, so,
Okay,
but
let
when
me-
we
I'll stop
started
there.
this, I don't think that was an option.
Okay, let me check on that.
Yeah. Okay. So as far as the pre-processing approach for the volumes, the fMRI volumes, it's probably fairly similar to what you've been hearing before, with a couple of additional things they've thrown in, mainly being the gradient distortion correction, which we're doing at the scanner, so that would be an option you would not choose to use as part of the processing tool. Motion correction, ideally using that single band reference image, otherwise you just use the first volume. Then the next step is applying the field map correction, followed by registering to our T1 weighted structural image, and bringing everything into MNI space. And essentially what the process does is it calculates what all these transformations are first, and then it applies them in one fell swoop to try and minimize how much translations and rotations and so on that you're applying to your data. And then it determines it masks the image so that you're now left essentially with just the functional image itself without any noise outside the brain, and then they normalize it, which is pretty standard for what you've seen with AFNI and other tools. And this is just kind of what the volumetric schematic looks like. And so as I mentioned before, all of these transforms are independently determined, and then they're applied in one step. So if you were using the pipeline itself, you would have to provide it things like what's the name of the different functional runs that you're using. They also want the phase encoding list. So they want to know what phase encode direction you're using. So HCP, with their protocols, a lot of their functionals, they do say a left-right, and then they do it again with a right-left. So I haven't bothered with changing the phase encode with mine, and that seems to have been working well. One of the problems that we've run into is when we convert our DICOM images using DICOM to NICS, or if we look at the output of that, I think we talked about this a long time ago, if you run DICOM to NICS, you get the image as well as a JSON file, which gives information about your data acquisition. In that JSON file, they have a variable that essentially defines the phase encoding direction, and it's incorrect for the GE. And this is a known problem to the developers of DICOM to NICS, and it's not something that they've been able to address. Again, I think it has to do with proprietary information. Let's see, if you have a single band, you tell it you have it, otherwise you don't have to define it. And we're using top up for our distortion correction. So in talking about the phase encode, you can kind of get that information to some extent. Sorry, I can't see what I have written here without my glasses. So since the HCP, thank you. Since the HP pipelines are developed for the Siemens, and they go off of phase direction, and we don't have that information. We have frequency direction as part of the readout from the scanner. So we had to kind of figure out what the appropriate phase encode direction was, and we partly did it by trial and error. And so what you can see here is that if we use Y plus, you can see that there's a bit of a different pull on the image relative to the Y minus. So the Y minus seems to be the right setting for us in terms of our data quality. Okay. Yeah, so you have to give it your field maps. We don't have gradient distortion. And then the last thing is simply the dwell time. So what's the sampling interval that was used for your data? So what I'm going to do is just go over the surface preprocessing next. So up till now, those steps are pretty similar, more or less, to what you typically would do to do volumetric processing. But since the HCP is very focused on using surface-based analyses for the cortex, there are this additional step that you would do to then bring everything into more accurate surface space. So the idea here is just that we want to take this volumetric time series and we're going to now map it into that gray ordinate space that Chris talked about last time. So as far as the cortex is concerned, you're using the white and the peel surface to kind of define where the fMRI voxels are within your cortical ribbon. Whereas with your subcortical, you're still talking about a volume. And what's nice about this SIFTY file format is that you can have both styles of information in the same file, so you're not juggling two different types of files. This script will also, in addition to identifying these voxels or gray ordinates, it will also resample your image and applies a light smoothing to it. And then at the end, combine everything into this single file format. So this is just what the kind of the processing looks like in terms of once you've gone through and you've cleaned out your volume in terms of putting it into M&I space, all that pre-processing, then we go ahead and we identify where our voxels are, we apply the resampling, we apply this 2mm smoothing. Meanwhile, we do some resampling and smoothing to our subcortical, and then at the very end, everything gets combined. And so just like with your functional data, you're just going to give it a list of the tasks that you want to run it on for each participant, what resolution you want. 32 is typical of the typical setting, and it's based on what value is used in the FreeSurfer pipeline. Excuse me, as is the image resolution itself for sorry, I swallowed wrong, for the spatial smoothing. And then we're using the MSM SOLC setting to allow us to register across the sulci. So at this point, if you've gone through these processing steps, we now want to go back and look at our data and make sure that the data look okay. We want to know, did the participant move a lot, or were they pretty steady? How much did they move? Is the data usable? And these things can vary depending upon, as far as how important they are to you, depending upon your population, and depending upon what your goals are with respect to the scan acquisition. So if you're doing healthy young adults, you would expect that they're not going to move a whole lot. But if you're doing kids or perhaps an elderly population, you may find that there's additional motion, and you have to decide, how much motion are you willing to accept as allowing that person's data to pass through. It also matters what task you're doing. So if you're doing resting state is really sensitive to motion. So you ideally want as little motion as possible. So that's one of the things you'll look at. Another thing is to look at how good did everything align. Is the gray matter surface properly aligned between your functional data and your anatomical data? Do you have any susceptibility artifact? Where is it? Do you care about that region where you're seeing the artifact? If you're not doing memory, then maybe you don't care if there's dropout in the temporal lobe. And sometimes is it so excessive that it's really going to affect your data? And so this is just an example of a plot that you can. I tend to generate these with AFNI, but this is an example of a participant who we took the output file of the motion regressors and looked at how much did this person move, and this person was actually pretty steady. There's a little bit of motion, but one of the things you got to pay attention to are the values on the y-axis, because AFNI, if you will scale it based on the data, and so if your voxels are 2 millimeter, but this person really moved less than 2 millimeter over the course of the scan, and this was task-based, I wouldn't worry about this one because it's pretty steady. But if these values were, say, +3 and -3, or +5 and -5, or if there's a head bob in there, then I would be more concerned about the data quality. The other thing that you can do is you can calculate the root mean square error, and that also gives you an idea over time about how much motion there was. Some people like to use the RMS value as a threshold for resting state fMRI, and I've seen different values used. I've seen some people use half the voxel size. Others are a little more conservative. So Van Dyke had a 3 millimeter voxel, but he made it very restrictive at 0.1 for the RMS. So I think, again, it's going to matter what population you're working with and what you think is acceptable. Because with one of our studies, there's no way we can do 0.1. The participants are just going to move too much. And so we have to just kind of take that into account. And so this is an example of what you can do in terms of looking at the alignment. So what's nice about using HCP and having the workbench application for visualization is that since everything is aligned, you can now get a really good sense, where's my mouse, of how well the functional image is aligning with the cortical ribbon here. And you can see that it actually does a pretty good job. We have a little bit of signal loss in the frontal cortex, particularly in ventromedial, which is expected, and a little bit here in the temporal lobe. But otherwise, this is an adolescent. Otherwise, this data looks really good in terms of the alignment. And I didn't have, if I had one, I would have showed you a really bad alignment, but I didn't have one come out on that I had access to. But I have seen cases where the alignment is really off. And again, that can be due to motion. So assuming that you've gone through all these processes of doing the volumetric and the surface-based steps, you could choose to just take those and move forward with your own processing, using AFNI's 3D deconvolve or using FSL feet to do your single level analysis. Or you can do some additional cleaning and registration of your data. And so that's what these next few slides are about. So as we know, artifacts are common. That's been, I think, hammered in. And despite the fact that we have advantages with using multiband, there's still some artifact that's going to occur. And if you're interested in doing functional connectivity analysis, that's something that you want to clean up because you don't want artifact to interfere with the relationships that you're trying to look at. So the approach that HCP uses is to apply independent components analysis as a way to identify noise. And I'm sure Frank went over ICA very well. I really like that talk, and explained how what ICA is and what it's composed of. But this is an example of using ICA as well as Fix, which is another tool that the FSL crowd provides, which helps to identify the noise components. And so what this is really showing you is an example of, at the top here, we can see an image that hasn't been cleaned up. And then at the second row, that same image where we've used ICA to identify the noise in our data and used Fix to help identify and remove the noise from our image. And similarly, these bottom two show something similar, that we have a noise, particularly around the edge of the brain, that's been largely cleaned up by using ICA and Fix. So what's nice about Fix is that it's an automated classification of these components. The software itself requires some training on a hand-identified data set. What's nice, though, is that if you're using the HCP protocol, they've actually already got a training data set there for you, and we're playing with that right now to see how well it works with our data. But it's been determined to be very accurate. If you have high-quality data, the argument is that it's at about 99% classification accuracy. That's probably on young, healthy subjects. So an important question is, well, how does this apply to patient populations? And I think we don't quite know yet. But even on a standard data set, it's about 95% accurate. And where did those slides go? They must be later. Okay. So with the ICA, it's combined with Fix as part of the processing stream. And what you would do is give it the names of your functional volumes. And probably the biggest question which we're now looking at is whether or not you should run ICA Fix individually, which means you do it on your monetary incentive delay task, you run it separately for the resting state, you run it separately for an auditory task, and so on. Or does it make sense to concatenate or combine all this information across down the time series? Does that give us more accurate data? And the HCP FSL people right now are arguing it may be beneficial to combine all of these time series, even though some of them are task and some of them are rest, because it's having more data and having more time points, it does a better job of identifying what the noisy signals are. So that, again, that's something that we're playing with right now to see how well it does. Trying to remember, I think one of the downsides is we have 9 functional runs total, and so we do have to be mindful of running that on our server, because if we try and run too many people at once, we get an out of memory error. So it's very intense. But if we run one subject at a time, it seems to be doing okay. And so you would provide that if you have multiple runs, it's a good idea to apply a bandpass filter. But you also decide, do you want to include motion parameters or not, what training data set you're using, and then there's a threshold that controls how sensitive versus you are. So there's a sensitivity versus specificity trade-off. So if you're too sensitive, you might lose something that's important, but if you're less sensitive, then you might be allowing noise in. Oh yeah, so I kind of talked about that. So one of the arguments that they make for why you might want to do single run is if your series is sufficiently long. So if you're doing a resting state scan and it's about 10 minutes or so, then you could absolutely do ICA fix just on that single run because it provides sufficient information. But if your series are all pretty short, it's advised that you concatenate everything. So, okay. So this is an example from the ICA fix that shows you what a signal might look like. And so what you'll see on the top left is this is the component spatial map that's displayed on the cortical mid-thickness volume. And then this is the spatial map that's displayed instead on a volumetric slice-by-slice basis across the brain. And then the bottom left is showing us the time course, and you can see it's kind of hard to tell because it's in blue. That wasn't me. But you can kind of see there is some regularity to it. And then on the bottom right is this power spectrum, which tells us how much variance exists at the different temporal frequencies. And what you'll find, okay, and over here, which if you were to open up Workbench and look at one of these images, you can actually select component by component and see each map individually, but it'll also tell you if that map was identified as signal or as noise. So you can go in and see what the different components look like and what they were labeled as. And what you'll find is that the ones that are more correlated with signal tend to have some brightness along the cortical ribbon. Whoops, where's my mouse again? No, there. Some brightness, typically bilaterally. And you might see it in the subcortical. And you'll also see that this signal down here, the frequency is a bit spread out, okay? As opposed to something that's more noise, in this case, you can see that the activity here down the time series is very rapid. And in this example, we still have this signal kind of in the same area, but we also have a little bit at the lower numbers and a little bit more at the higher. So this tells us this is probably a noisy signal. I think, there's a couple of really nice papers that were put out by this group, one talking about hand classification and gives lovely examples of the different kinds of components you might see. And then there's the second paper where they talk about fix, and I think I referenced it. Yeah, these two papers that talk about ICA and how it does on your data. Okay. So after running ICA and identifying the noise, the next step is now to align your data across all the data that you've acquired in a more refined way. And so there's an algorithm, MSM-all, which is this aerial feature-based registration that will use all of the data that you've collected for your participant. It uses the T1 and T2 weighted myelin maps. It uses resting state, task-based, all of this information to align your cortical data to the group template. And what the argument is that the HCP crowd is finding that it improves your overall cluster identification, the cluster mass, as well as being beneficial for distortion. And so we've run it, we're just starting to run it on some of our participants now, and we're finding that it does a really nice job of refining the alignment, because what it's now doing is it's using the surface-based mapping to do that alignment as opposed to more of a volumetric approach. And the big key with using MSML is that you're going to use the same, you identify your parameters in the same way that you identified them for ICA fix. And so if you've run ICA fix just on a single run, you're going to want, or let's say you have 5 runs, but you run each one individually, you're going to need to run MSML individually on each of these runs. If you've done concatenated, then you can actually just tell it all of those images at once, and it will take that into account when it does the registration. And what it will do is just output what the appropriate registration values are, so that the next step, again, this is now taking those parameters that you've identified and applying the resampling to your data. Just as we've done that early on with the volumetric step where you figure out the transform and then apply it, it does the same thing here. The de-drift, that's not something that we're going to use, so we don't need to worry about that. It's mainly if you wanted to generate your own atlas. We're not going to do that. So one of the heads up is about HCP processing, is that it takes a while. So if you've ever used FreeSurfer, depending on the server that you have, it's going to take anywhere from 8 to 12 hours per subject to run. The diffusion pipeline takes about an hour. Your fMRI going through that volume and surface-based processing takes on average 5.5 hours per series. So if you've collected 8 series, it's going to take a while. ICA fix takes at least an hour and a half. With our study, we have 8 tasks and 8 task runs and a rest. It takes about 6 hours to run through ICA and fix. So it doesn't quite scale if you were to concatenate, but it does take a while. And MSML, again, it takes about 3 hours to calculate, but then the de-drift resample step takes about 15 minutes per series. So you're looking at least 24 hours just to process your subject. But hopefully the data is of better quality at the end than some of these other tools that you've used. And isn't our data worth it? So at this point, you've now gone through these headaches of doing all of the processing, and now you want to analyze your data and you want to take advantage of all of the HCP steps that you've done. And so for the task-based approach, it's pretty similar. If you've ever used FSL, it's pretty similar to FSL in that you're using FSL's GLM, their general linear model tool, to run it on your first level design. So you would run each run individually and then create a second level to look at the higher level run, the higher level design. So if you had four of your tasks, you would run those individually, and then at the second level you develop your contrasts to look at the participant across all of those series. And then from there you would use those contrasts that you've generated for each subject, and you would run them through FSL's PALM, which stands for Permutation Analysis of Linear Models, which is a non-parametric permutation method that will go and test your linear hypotheses. We've just gotten this to work and are just starting to work with this now on the Phillips data. What's nice is that PALM not only takes NIFTY files, it'll take the SIFTY files as well. So I've used it and I've seen it used on myelin maps. If you wanted to look at your control group versus a patient group, for example, you could run it on the myelin map and see if there's any differences between your groups. And this is just an example of what your between subject analysis might look like. And so in this example, this is looking at a contrast related to a lower level working memory task, and looking at whether it's associated with a delay discounting variable as well as the ability to store and sort through lists in working memory. And so what's nice is that this shows us a couple of different clusters that identify these working memory related activity that's correlated with our two different designs. And so, whoops, there we go. So in this case, this one is correlating with the ability to how well we or don't perform delay discounting. So how much do we value a delayed reward relative to immediate reward? And then this cluster is related to an association between the working memory and the ability to sort through our lists. I have not played with this at all yet. I'm just starting to look into this one, but if you were to do task-free analysis, you can take the output of you would use Melodic to do your group level ICA to generate your group level spatial maps, and then you can essentially make these network models of the time courses to look at your resting state data to kind of do some network modeling on that. And FSL has this tool called FSL Nets for that. I haven't played with this. That's on the list. But what's nice is that it allows you to kind of look at the correlation strength between your two nodes. And so this is an example of a network where they found a relationship between fluid intelligence and some resting state data. Whoops, that's right in here. So looking at the measured fluid intelligence versus the predicted fluid intelligence based on the resting state data. Okay. So again, why do you care? So if we start with just looking at our myelin map, in this example, we have this little strip here that's lightly myelinated between the frontal eye field and the premotor eye field, okay? And we kind of see that there's these bright spots here to represent other areas that are areal boundaries to that little strip, okay? And if we do task, in this case, it's a task where the participants are presented with a story relative to some baseline condition. We find that task fMRI actually lights up right here in the exact same spot. Now, if we do functional connectivity, we can now see, with respect to different seed regions, that we have this seed in the interparietal junction that seems to be strongly associated with that region of interest. Whereas if we have this seed in the parietal cortex, we don't see that it's related here, but we do see that seed is related to the regions that bound it. Okay. And this is basically, so now we've got all these different modalities that are agreeing or to identify this region of interest. And this area is identified as area 55B in the HOP parcellation. So what's really nice is now you've got all of these different modalities that kind of help you better identify or refine where your activation is for your task or your connectivity measures, because you're taking advantage of all of the different modes that you've collected. So there's a lot of software that's involved with HCP. I mean, first and foremost are the pipeline scripts themselves. But then there are all the tools that it depends on. And these include things like FreeSurfer, FSL, the Connectome Workbench, which is useful for visualization and discovery. So that was what I used for some of the imaging displays. And it works really well with looking at data across multiple modalities. Fix, which is that denoisifier tool by FSL that is meant to auto-classify your ICA components. Although it's by the FSL people, it's a separate install and is dependent on using MATLAB or MATLAB Runtime or Octave for some of its processing, as well as a number of R libraries in R. I can tell you that it is a pain in the neck to get Fix to run initially. Fix is very brittle in terms of its software dependencies, and we have found that if we don't use the exact library that is recommended in R, it doesn't run. It will tell you specifically which, if you're using runtime MATLAB, which is actually what they recommend, they will tell you which version to use, and it won't work outside of that version. So fun times with that. MSM HOCR is that multimodal surface registration package, and it's different from the one that comes with FSL. So it's a little bit more refined, and that's the one that we use as part of the pipeline. And then at the group level, you have Palm, and you also have FSL Nets as two possible packages that you might want to look into if you wanted to take advantage of all of the processing work that you've done to this point.
But does the HCP provide a Docker container or anything that has everything installed that you can just...
There is a Docker container involved. I don't use Docker. But I don't know if it's developed by them specifically or another group. Based on what I've read on the HCP mailing list, it seems like most people install the tools individually and then complain when they don't work. So, yeah. Oh, and that's another thing I should mention is that they do have a mailing list. The mailing list is a little frustrating if you're used to searching archives, in part because they've shifted everything over to Google groups. And so what I have found is that just looking there is one area where there's HCP archives outside of Google, but that is old by at least a month. So if anything has happened recently, you miss out on that announcement. But the Google group is the one that actually has everything up to date. So if you're interested in using HCP, I would highly recommend attending the workshop. It's a 1-week workshop. I haven't seen an announcement yet about 2020, but it's typically held close-ish in time to HCP. In past years, I think it was like the week after HCP, but more recently they've been, or
HBM.
Yeah, HBM. Thank you. So yeah, it used to be, I think, like the week after, but lately it's been about 2 or 3 weeks separated out, and it's not necessarily in the same place either. So that's all I have on this. Are there any questions? It was probably boring, but...
Is there any sense of whether the HCP and the ABCD processing pipelines are going to have any similarities?
So that's a really good question. ABCD, from what I can tell, seems to have adopted it partially. So there are some scripts on GitHub by ABCD which seem to take advantage of the HCP processing tools, but they're not all there yet. I've seen them mainly replicating the structural side, but less so on the functional. But I haven't looked recently, but that was a few months ago was the last time I looked. So Are there any other questions? We're done really early. Everybody's probably glad. It's time for a nap. So y'all ready for your presentation next week? No.
So I sent out a corrected schedule, so if there's any mistakes in that, let me know, we'll send out a second corrected schedule. What else? So yeah, please fill out evaluation and leave them back here.
Yeah.
Dr. Gress. We'll see you
Yeah.
on Wednesday.
Yeah, and we'll be posting questions within the next 24 hours, the quiz
Right.
related to last week and this
Right,
week.
to the final, so there will be a final quiz related to the last two HCP lectures. That'll be due, I guess, Friday, right?
We'll make it do.
Excuse me?
That's what Chris said it would be.
Okay.
Yeah, yeah.
So then, and also if, I think most of things have been graded, but if you look on your grade sheet and there's something that seems to be missing, just send me an email and we'll figure it out. 'Cause we have had multiple people doing the grading, so there could be an issue, but we fixed some issues today. What else? So then the final project presentation, and then there might be a few more, ungraded quizzes during the lecture on Wednesday, and then I'm also going to be making up sort of a survey for sort of course suggestions, so one less ungraded survey. So I'll announce that on Wednesday as well. Okay, any other questions?
I think the lectures from last week are not uploaded.
There is, the DTI one is now uploaded. The HCP one from last Wednesday, there were the technical issues, so Chris is going to re-record that. And hopefully this one was recorded today.
We'll find out.
I've been trying to get Dr. Heist to give me his videos, but we're not really sure, so those may not